I know the topic may be a little bit little far from what you are doing, but I hope it is related in some sense, and probably we can find some common ground that we can discuss today. Yeah, so thank you for the nice introduction. I'll be talking today about some work that we have done in the last few years and resulted in a publication in Nature. But the main topic today is to talk about machine learning and how to make it more interpretable. So let's get started. If you have any questions, I am happy to answer them. Just write them in the chat or somewhere else and just keep them to the end. I'm happy to answer any questions, and even if we didn't have like enough time today to discuss everything, probably we can find another time to discuss offline. With this, let me share my screen. I hope it works this time. Every time it makes some, yeah. I hope you can see my full screen. Okay, perfect. Sounds good. Let's move this here. Started. Okay. So what's machine learning in general? Or first, we are talking about.
We are seeing the launch meeting Zoom.
Okay. Okay. What about that?
Now we see your slides.
Okay, perfect. Sounds good. Sorry about that. Just the two screens issue. Okay. So we are talking about machine learning. So what's learning, first of all? So we are talking about learning the machine, or the machine being able to learn. So what's learning? So for any system to be able to learn, it means that it can improve its performance from experience. So if a human can learn, which means that he can enhance his performance or his skills by learning new things. The same applies for machines, and we'd like the machine to be able to learn from experience, and it increases its performance over time when it is like provided with multiple experiences. So we'll talk about this in a second. So if you think about any computerized system, any system that is that involves some computations, it is mainly you have some system, like some machine in the middle, in the core of the system, and you have some rules or programs. You have some inputs that can generate some outputs. If you think about any computerized machine, like financial system, like a website or whatever, you have some machine in the core. You have someone actually who knows how the system works or how it is supposed to work. And this one actually, he or she can be a programmer that can write the code inside the machine. And this machine can handle any kind of problems that are for which this machine is designed to solve. So if you think about financial system, the input is like a bunch of numbers, and the output may be like some reports or something like that. But we have someone actually, some expert in the middle who knows how the system should work, and he or she like will write some piece of code that can handle different kinds of inputs and generate the desired output. Okay. In machine learning, we are trying to solve the inverse problem. We still have a machine in the middle. We have some inputs, but we don't know exactly how this input will generate the output. We don't know the essence or the way the program should be working. So in machine learning, we are trying to teach the machine actually to come up with its own program, with a program actually that can generate outputs. But before generating any outputs, actually, it should learn first. It should be trained first. It should be seeing multiple problems and how these problems can be solved, and then learn from these experiences how to generate this model or this program. So instead of hard coding the program, instead of having someone sit down and writing all of the rules to solve a certain problem, we have the machine actually trying to figure out how to solve a certain type of problems by providing multiple instances of the problems and solution to this machine. And hopefully over the time, through different training algorithms, the machine itself will be able to solve this problem, but not only to solve the problem, but also come up with a model or a program that's a little bit different than the programming language, program that can solve any specific instance of these problem types. So in machine learning, it's a little bit different than the typical programming language. This is different from any typical information system that you are trying to build. And applying this actually in the cancer space, we have so many data types. And here probably you can find some commonalities between the thing that we are doing here in the lab and things that they are doing over there. So we process a lot of genomics data, and this genomics data can come from somatic information, germline data, whole exome sequence, panel sequence, or any other kind of sequencing that generates genomics for either patients or cancer cell lines. We also do a lot of gene expression data. So we work with a lot of transcriptomics data, whether this is coming from bulk RNA-seq or single cell RNA-seq or even spatial transcriptomics. We work with sequences like DNA sequence or protein sequences. And we work also with images, whether this image coming from X-ray, from MRI, or any other like imaging techniques out there. We work with texts as well, whether this text is coming from medical reports or clinical notes or history of the patient, things like that. So as you can see, we have a lot of data types that we can handle. And our lab actually works with a fair amount of these data types to answer different questions. So we are trying, for example, to solve the problem of predicting clinical outcome for patients. We are trying to stratify patients based on different risk factors. We are trying to come up with grading systems or subtyping systems for different patients. We are trying to answer questions like why a certain patient is responding to certain drug while other patients are not responding to the same drug. And can we find markers for doing this? And actually the data comes from a lot of sources, but our focus in the lab actually on the clinical data. And we have a special interest in genomics, transcriptomics, text, and imaging for different reasons. Of course, there are other data sources in the cancer space coming from cancer models like cell lines or from xenograft, from mice models, from organized, whatever. But in our lab, since we are based inside the hospital, we are focusing more on the patient's data. And cancer. So the current understanding of cancer is that it is a genetic disease, which means that a normal cell can acquire a certain mutation, and this mutation comes from whatever source, like from radiation, from chemical exposure or whatever. And probably the offspring of these cells will have the same mutation as well. And maybe even like some of these offsprings may acquire more mutations. And the accumulation of these mutations over different, like multiple generations actually can lead to malignant cells arising in certain part of the body. And this tumor actually forms the basis of the cancer. So as you can see, we have a lot of events happening before the cancer happen. And we are trying to understand actually what is the relationship with of these mutations, which these mutations is important for predicting different outcomes. So this outcome can be drug response, can be survival for the patients, can be progression of the tumor over time. So we are trying to find a relationship between different mutations that happen in the tumor side and the clinical outcomes of the patients. And hopefully this will enable us to do a lot of things like change the clinical care of these patients, early monitoring of these patients, stratifying patients based on different risks. So we can do a lot of in the clinic, but if we know, if we got a patient and got some sequencing coming from these patients, and we were able to analyze these mutations and identify subset of these mutations that have like clinical effect on the trajectory of the patients over time. But there is a problem actually with just working with the individual mutations, because actually there are a lot of mutations that happen in different parts of the genome. So we have a lot of genes, and these genes actually are susceptible to different kinds of mutations. And not all of these mutations are actually important. A lot of these mutations are like, I mean, there is no function for these mutations whatsoever. And even some mutations, probably you can think about different mutations may have similar effect. They are like leading to the same protein or something. So for example, this is an example from a data that we published like a few years ago. We collected a lot of prostate cancer patients and we tried to see which genes are mutated. And as you can see, most of the genes here actually are mutated in very low rate, like 2, 3, 1% even, with the exception of TP53 and SPAW. So TP53 is known to be mutated, highly mutated in different cancer types. So this is not a surprise. But most of the other genes actually are mutated in very low rate. And frankly, we didn't know exactly what to do with all of these genes and how to rank them, or what to do with all of like hundreds of genes that are mutated in very low rate. So we need some way actually to let us know which genes are really important and probably may have a clinical impact on the patients. Of course, actually, we cannot build our decisions based on all of these genes. We need like a subset of genes to better understand the function of these genes in the tumor progression and the drug treatment. So one solution here actually is, instead of like thinking about the level of the genes, to think about the level of pathways. So pathways, like a set of genes, are working together to function in some way inside the cell. And this is like an example of different pathways that are important in cancer. So probably you can think about the group of genes instead of like individual genes. So first, first of all, we were thinking about individual mutations, then we moved to individual genes, and later we moved to group of genes. And the idea here is that we are trying to gain some more power, because actually, if you have like a feature for your patient that is like only exists in very small fraction of patients, it is not very informative in deciding the outcomes, in informing the outcomes. So probably you are trying to gain more power by aggregating these features on different levels. So the level of the individual mutations, the level of genes, and then probably the level of group of genes that work together actually to achieve certain function inside the cell. So I'll keep this for a second. I'll come back to it again. But if you have like a prediction problem that you are trying to solve using cancer data, you have a lot of options in machine learning space. So you have a lot of models out there. You have like simple models like logistic regression, decision trees, or even more sophisticated models like deep neural networks. And each one of these models actually come with its trade-off between accuracy and interpretability. Simple models like logistic regression, they are very simple. They are just a bunch of weights for different features, and they are very, they are very interpretable. I mean, you can just inspect these weights and come up with some understanding which feature is more important. But usually in solving machine learning problems, these models are not very accurate. So the computational performance usually actually is lower than the other models that are more sophisticated. But the problem with the sophisticated models is that they are not very interpretable. We don't understand exactly how they are working, what is going on under the hood. So what we are trying to do in this project is that we are trying to enhance the interpretability of these sophisticated models. So it's not enough for biologists or for like clinicians to let them know that here is the pipeline, we put a machine learning here in this step, and some miracle happen, and this is the outcome, right? We need to understand what's going on. And it is not only about solving a prediction problem, but rather it is more about understanding mechanisms in the cancer space. So we are trying to open the black box of the machine learning model, and probably through different techniques like visualizing different parts of the model, inspecting different characteristics of the model that we trained, using different attribution methods, we are trying to open the box and see what's inside to some extent. We are trying to increase the transparency and interpretability of the models. And the word interpretability itself is like a vague word. So it is, when we say interpretability, usually we think about conveying something to the end user other than just a bunch of numbers. We are trying to give the practitioners or the biologist or the clinician something to relate to and something to understand. So for example, if you are working with the space of the images, probably you can give the end user some images. So instead of giving him like a bunch of numbers or telling him that we train like this convolutional neural network to predict the outcome, we can give him like some parts of the model visualized in some way that is similar to the input space. So if the input space is just some images, probably we can give him some images as well to see which part of the image actually this model is focusing on at this point, and which part of the model is related to which part of the input image. So things like that actually may enhance the interpretability of the model and give us some understanding how the model is behaving. So for example, in this slide, you have the model actually focusing on like edges and texture in the far left, and probably other parts of the animal in the far right. Yeah, I know it's a little bit messy, but you can see that there is a scale change from the input to the outcome, and also you have some different focuses from the first image to the last image. So here it is another kind actually of interpreting the model, where the input actually is just a bunch of images, and the outcome is some kind of prediction, but also some kind of like heat map, where actually we are trying to focus on the parts on which the model actually is attending right now. So the model here is focusing on the birds or the animals. So this let us know that the model actually is processing these parts of the image in a different way other than the background, right? So the model actually is focusing on something that is reasonable in terms of the prediction. So what we are trying to do is that we are trying to enhance the interpretability of sophisticated models, and in this case it is deep neural network, but we are trying to get the best of the tools, like getting good computational performance, but also getting better interpretability. I'll go just a little bit with few things about the architecture, but if you think about a simple model, like a logistic regression model, it can be visualized like this. You have a bunch of inputs, x1 until x6. These are the input to the model. You can think about these inputs as anything, like sequences or mutations, mutation count per gene or something. And you have some kind of output. Probably this is drug response or whatever. And the relationship actually between the input and the output is just a very simple equation like that. It is just these features multiplied by a set of weights. And this is just a very simple linear or logistic regression based on the function f. Right? So we are just multiplying each feature by a certain weight, summing up all of these weighted features, and feeding this into a simple function, whether it is linear or sigmoid function, to come up with an outcome. So this is a very simple linear model that you can implement easily, can interpret easily. But if you like to have like more sophisticated models, instead of having one layer here of these weights, probably you have multiple layers of these weights, right? So you have your inputs as well, but you have multiple layers in the middle that can do multiple transformations on the input until you come up with the outcome at the end. So the nice thing about this model is that it is able to capture signals from the input accurately, but it has a problem that we don't have actually a lot of guidelines of how many nodes should we include in each layer, how many layers should we include in the model, what is the kind of connections between these nodes, or what is the function that each node actually is achieving inside the model. So whenever you have a problem, you start with arbitrary architecture, you tweak and tune it over and over and over until it is able to fit the data. What we try to do in this project actually is that we try to come up with like more informed model instead of using like arbitrary layers, we try to make sure that each layer has a meaning that is related to the biology that we are modeling. And each node has a meaning actually that is related to the features, genes, or pathway that we are trying to model as well. So here, as you can see, we have set of inputs, and these inputs actually represent set of features that we are able to measure experimentally. These features can be mutations, copy number amplification or deletion, gene expression, methylation, or whatever feature that we are able to measure in the lab. And then these features actually are feeding into a layer of genes. So each node in this second layer actually represent some kind of gene. So if you have like set of genes that you are interested in studying, you put them here in this layer. And then these second, third, and fourth, and fifth layer, they aren't shown here, but we have multiple layers out there. They represent different pathways. So if you remember, the pathway is just a set of genes. So if we know that a certain gene is a member of a certain pathway, we add a link between this gene and that pathway. And the connections between all of these nodes are like based on our prior knowledge that this gene is part of this pathway, or that pathway is part of a super pathway, or that super pathway is part of this biological process that you are trying to model. I'm not going to in deep details about the model, but if you think about it, I mean, it seems simple here, but what we did is that we took all of the reactive data set, like more than 2,000 pathway with associated genes, like 20,000 genes, and we included all of this into the model itself. So the model at the end actually is based on our understanding of the biological processes. So each cluster of nodes here actually represents some biological process, for example, DNA repair, or like immune system, or cell cycle, or whatever. And each process actually includes multiple pathways that has multiple sub-pathways and so on and so forth. And each of these leaf pathways, they have genes associated with them as well. So we took all of these curated pathways and we transformed them into computational model that is able to receive inputs and generate outcomes in a neural network language. So it is like a kind of taking all of these things that people worked on for decades, trying to create all of these pathways and their relationships and their hierarchies, and put them into a neural network architecture that respects the biology included in this hierarchy that we see in front of us. So on the left here, it is our model. We call it PNet. I don't know the abbreviation, but probably it's pathway network or whatever. And on the right, it is the off-the-shelf neural network that everyone uses. As you can see on the left, actually, we have some meaning for each layer, some meaning for each node. The connections between the layers are based on our prior knowledge. On the right, actually, we don't have meaning for the nodes or the layers. The connections are like dense connection, which means that each node is connected to all of the nodes in the next layer and the previous layer. So on the left, actually, we have some architecture, some guideline to build the model. On the right, we don't have such guideline. And what's more important, on the left, actually, we are able to interpret the model in a very informative way. On the right, actually, while we still able to interpret the model, we don't have a meaning for our interpretation. For example, if I told you that there is a node on the right, actually, that is important, you don't know what this node actually is representing in the real life. Like, it is just a computational node in the middle of a computational model. But on the left, actually, it is a computational node that is related to some biological entity that we are interested in, whether this is like a function, feature, or like a gene, or a pathway, process, or whatever. So this is about the model that we developed, and probably I'll give you some context about the problem that we solved. So we previously published like more than 1,000 patient whole exome sequence sample, and this data is available on CBio portal. You can go download it if you like. And these patients actually, some of them are like taken from a primary site, and some of them, they have metastatic cancer. So we took the samples from the metastatic site. These are like prostate cancer patients, and usually the metastatic site, actually the prostate actually goes through the bone marrow. So most of these patients, they have like bone marrow cancer. So we try to see if there is any genomic differences between the two subpopulations that we are interested in. And can our model actually capture these genomic differences? And can we interpret this model in a useful way that actually gives us some understanding of the differences between the two subpopulations? So the input to our model is a bunch of mutations and also the couple number information, whether a certain gene is deleted or amplified. We divide the data into training, validation, and testing, and we got some results. Here, for example, our model is correctly identifying 93% of the primary patients. We compare the performance of our model with other models like support vector machine, risk regression, things like that. Our model is achieving slightly better, well, it's better performance at 0.93 area under curve. If you do like the evaluation on the cross-validation setup, which means that you take the data, divide them into different folds, and train on 4 folds and test on the remaining 51, and do this like multiple times, we have that our model is still achieving the best performance using area under curve, area under precision record curve, and even the F1 score. These are like just computational metrics. Also, we compared our performance with typical dense network with a comparable number of parameters. We see that our model is achieving better performance. And down here in the right corner, where our model is shown in blue and the dense model is shown in orange. We did also like external validation. We found that our model is correctly identifying 80% of the metastatic patients. And these are like data that the model was not trained on. These are like external validation data. We also tried to identify the modes of failure of our model. So actually our model is not perfect. It makes some mistakes as well, but we tried to see if these mistakes actually is are informative in another way, not only in the predicting the right outcome, but also can we make use of these scores coming out of the model in another way. So we found that the model actually that identifies, well, patients who received like high score from our model, which means that our model thinks that these patients are metastatic patients. However, they have primary tumor. So this is definitely wrong prediction of our model. But we found that these patients actually, they have like worse prognosis, which means that they will die faster than other patients. So this means that our model actually is, well, probably capturing something there. And probably this score this score that is generated from the model can be used in another side problem to solve actually, which is like stratifying patients based on their risk of death and even before actually having their tumor metastasized to the other parts of the body. So this can be, so this may indicate that the model can be used in early detection or early stratification of patients. We try to interpret the model. We are trying to predict the outcome, which is which patient has primary and which patient has metastatic tumor, but also we are trying to understand why. What is the difference? So we found that the data type that is more informative in our prediction was like couple number amplification compared to the mutation and the deletion. So this means that the progression in prostate cancer is mainly driven by couple number events in the patients. This was interesting. But also we tried to see which genes actually are amplified or mutated or deleted that actually affect the differences between the primary and metastatic tumors of these patients. We found things like AR, TP53, PTEN, RB1, MDM4, and other things. And we tried to see, well, so if AR is important, why is it important? So we can, using some normalization and processing here, we can see that AR is mainly driven by amplification. So which means that we have a lot of amplification of gene AR in prostate cancer, especially in the metastatic patients. For the TP53, we found that mutations of TP53 is important in predicting the outcome. In PTEN case, actually, the gene PTEN is deleted a lot in the metastatic patients. RB1, it is either like mutated or deleted as well. So these genes, I mean, the first 4 genes, at least they are known in the literature. So our model is able to capture known knowledge from the biology of prostate cancer from the data, without a lot of expert interventions. However, we found that MDM4 actually is highly ranked. It's not very known in this situation. So we tried to follow up with MDM4 because it is interesting. And this is surprisingly highly ranked in this setup. If you just continue with the interpretation thing, actually you can get some image like that, which is like each node, the importance of each node, the size of the node actually represent their importance and why they are important and their relationship between each layer. So this can give you like a multi-level overview of the system that you are modeling in terms of features, genes, pathways, and processes. Right? So MDM4, well, it seemed that not only MDM4 was important in this setup, but also it is important because it is related to TP53 pathway. So we have a lot of TP53 pathways like highly ranked in this model. And as we know, actually TP53 is a very important gene that actually like regulates a lot of processes inside the cell. But not only TP53 is regulating a lot of processes, TP53 itself is highly regulated gene, which means that there are a lot of mechanisms to regulate TP53. So if you are a cancer cell that you are trying to get rid of TP53 because actually it is regulating a lot of processes, you have so many options. So you can introduce a mutation inside TP53. You can delete the gene TP53, or you can up-regulate some of the negative regulators of TP53. So we have like negative regulators like MDM2, but also MDM4 is a negative regulator of TP53. So if you if you increase expression of MDM4 or amplify the gene MDM4, this will down-regulate TP53. So this will actually affect all of the processes, the biological processes of the cell. And it seemed this is what was happening in this situation where MDM4 actually was amplified in a lot of prostate cancer patients, especially the metastatic patients. So we tried to see if MDM4 actually is mutually exclusive with other genes, or what is the relationship between MDM4 and AR and TP53. We are interested in AR and TP53 because they are like well known in prostate cancer, that they are like very involved in the biology of metastatic patients. We found also that MDM4 actually came up in another cell line screens, cancer cell line screens from one of our collaborators. So they did like ORF screen, and they found that MDM4 actually is highly associated with enza resistance in cancer cell lines. So this was interesting that we have two sources of information, one coming from the computational modeling that we did, and one coming from the experimental work that other collaborators did in the cancer cell lines in the dish. So this was interesting that MDM4 may be important. We followed up with more experiments with MDM4, and we did like CRISPR knockout for MDM4. We did like also drug inhibition for MDM4 and MDM2. We found that cancer cell lines actually that receive MDM4 inhibition, they are more sensitive for the treatment. They die like much faster than the other cancer cell lines, especially if this cancer cell line has a TP53 wild type, not mutated. So this was interesting actually to start from data, like data collection, to machine learning modeling, to interpretation of the model, and then to follow up studying and experimentation with one of these genes to the discovery that the gene may be important in the treatment response, especially in the cancer cell lines, and this may in the future actually translate into the clinic. So this actually story from the data to the experimentation to the validation is an interesting story actually that we reported in this paper. And we hope that using such model, actually machine learning model that has a lot of interpretability characteristics, that we will be able to solve other problems as well. So right now, actually, we are working on some of data analysis for immunotherapy response prediction. So we collected like a large cohort of patients who received immunotherapy treatment, and we are interested to know if there is any markers, biomarkers for treatment response that we can discover using these novel machine learning techniques. We are working with our collaborators at MD Anderson, and we hope that we can find something as well, but it depends on the resources like the students and the time that you can dedicate for such a project. So with this, actually, I'll conclude my slides. I took like exactly 40 minutes. I hope that you will find this interesting, or at least this may initiate a kind of conversation that we can follow up with. I'm happy to receive any questions if anyone has any questions.
So thank you, Aitam, for your nice presentation. And as you've said, now we can ask the questions, and discussion is now open. So feel free to ask your questions and your comments using the raise your hand function of Zoom, or write them directly in the chat box.
Yeah, we can start with the question in the chat right now from Dr. Ahmed.
Yeah. Do you want to ask the question yourself, or you want us to read them?
Okay, as you wish. So based on what you wish, I can verbally say my question. So I have a question. So I have many questions. In fact, I have sent also many questions to Benjamin directly. So my first question is that in your experience, Dr. Elmarakeby, what determines the size of outputs that can be used to train a model? So what are the criteria that helps me determine the size? Because usually this is something that should be considered before starting to use machine learning.
Yeah. Yeah, if I understand correctly, this is about the amount of samples that we need to train any model, right?
Yeah.
I would say this is the most difficult question anyone can answer. It is very hard. Well, at least in the statistics field, they have what they call power analysis. They can do like power analysis to identify how many samples they can they need to have to get like confidence in the outcome, right? But this is done actually because they are using only like one variable, or at least if they have like multiple variables, things will become complicated. But here in the machine learning space, we have like usually we have like hundreds of thousands of features, and usually we don't have enough samples. In any machine learning models in the medical space that I worked on, we don't have enough samples. But at least you can do some things with the data that you have. So there are multiple techniques out there, like things like regularization that you can try to use to computationally select subset of these features instead of using all of the features, or things like dimension reduction, where you're trying to like find another representation of the data in a lower dimension space. So instead of using like 1,000 features, probably you can use like 10 features, like from PCA or from other like dimension reduction techniques. And all of these techniques, none of them are perfect. None of them are perfect. But people are using them just because actually they need to use something, and they need to do some analysis on the data. And there are other metrics actually to see how we are doing in terms of prediction, whether your prediction is stable or not. If you use like multiple different samples, when I say samples, subpopulations of your samples, like divide them in different ways and see if you are getting consistent results or not. So this can give you some indication that your model is doing good. It is stable and consistent over different subset of your samples, things like that. But none of these things are perfect. And the short answer is that the more the better. So if you have more samples, this would be better.
Yeah.
But usually you don't have the exact number. If you have like 1,000 patients. So we in our case, actually, we developed this model using 1,000 samples, right?
Yeah.
You can expect like similar amount of samples should be fine. Yeah.
But in fact, so let's assume that we started with a specific, with the data that we have, number of outputs that we have, and how shall we know by the end of the model that it is the number of samples were sufficient or not? So for example, shall we see or examine the accuracy of prediction or power of the model? So what will tell us that in fact, by the end of the artificial learning pipeline, working pipeline, the number of samples were insufficient, for example, or we need more samples. So what metric will tell us, assuming that we work along with the whole pipeline, with the number of samples that we have, for example?
I see. Yeah, so there are some techniques actually to do this. Like there is like a best practices in machine learning field where you divide your data into training, testing, and validation. Even the training set, you divide it into multiple cross validation and like evaluate the performance of the model on different faults or different subpopulations, making sure that your model is consistent over different samples. So what happens if you don't have enough samples, that probably your model will be behaving very well on a subset of the patients, but very bad on the other subset, because actually you have a lot of heterogeneity in your data that this sample, this number of samples aren't capturing the whole story of your data. So you will find this inconsistency between different data splits in your patient. And you can identify it very quickly, actually. If you just train a couple of models and compare their performance, you'll find that they are not very consistent. Or the worst actually, that you find that your model is not able to fit the data, which means that it's producing a very bad results from the beginning, because actually the signal is very diluted actually, or the spread all over the data. Actually, the model is not able to capture it. Or there's no signal at all, actually. Probably the assumption is that there is a relationship between genomics, for example, and certain clinical outcome. Probably this assumption is not a valid assumption either, because actually there is nothing, there is no signal there, actually. Probably the signal is somewhere else, not in the genomics, maybe in the transcriptomics, maybe in the protein abundance, maybe in the pattern of distribution of the proteins. So it can be somewhere else, maybe in the epigenomics, right? It doesn't have to be in the genomics. So yeah, so you can find a lot of things, but probably you will not know until you try. Probably like a couple of trials should be enough to tell you about the complexity of the problem that you are trying to solve.
Thank you so much. Thank you. Fair enough. Thank you so much. Do we have Benjamin? Do we have other questions? Otherwise, we can.
No, I don't think we have other questions more. But just I had one question I think will be answered very quickly. So basically, from what we have with the machine learning tool, we could, for example, sequence genomic DNA of a patient and then run the tool and be able to find if the person is predisposed to certain types of cancers, and which treatment could be the best to use depending on the patient. and his genomics data.
Okay, is this a question or yeah? Is this an assertion or question? I'm not sure.
No, is it something we could do with the prediction tool at the bottom of the line?
Yeah, so just to let you know, this is not like a ready-made tool. It is a research project. So a ready-made tool will be like more usable and easily accessible. But this is what we have done is more of a research project that has a lot of stakeholders, and the end result actually is it is available on GitHub if you like to download it, but it's not like a tool that you will download and feed something and get something out of it. It's more of a development cycle where actually we were talking to so many people, like many stakeholders, computational, clinician, and experimentalists who have different views, and they contribute a lot to the model development over the time and the interpretation of the results and the fine-tuning of the manuscript, all of these things. So I would say it is like a little bit more involved than just like a tool that someone can use it. What you described actually is our dream, that someday in the future, hopefully the near future, we will be able to sequence the data of a certain patient and get a lot of information from this sequence. Like, okay, this sequence will tell us a lot about the future of the patient. It's not the case right now. It has like limited success and limited cases of patients who have, for example, certain mutation in certain genes. We know for like with good confidence that if the patient has this mutation in this gene, he will be very responsive to certain treatment. This happens in breast cancer, in prostate cancer, and in other cancer types, in lung cancer with target therapy, things like that. So, it has like limited success in certain subpopulations. Discovering these subpopulations who are like very responsive to certain treatment is something that we are trying to do for each cancer type. Right? So we are trying to do this with the immunotherapy project that we are trying to develop as well. We are trying to find a subpopulation of patients who can respond to immunotherapy with good confidence. I would say that there are a lot of challenges. If you asked me like 5 years ago before joining Dana-Farber about certain problem, I'll tell you, well, give me the data, I'll solve the problem for you. It's not the case, right? It's very, very much more involved than that. There are a lot of challenges. I can tell you, like, I can talk to you until the morning about the challenge that someone can face, starting from the data generation, from the assumptions made in the QC and quality control and the data pre-processing and data harmonization and machine learning modeling and interpretation and experimental validation. Everyone comes with some assumptions and some shortcomings. It is not perfect at all. It is a huge pipeline, and we are hoping to like enhance each component of this pipeline over time. But yeah, it needs a lot of time and a lot of like smart minds to contribute to the solution of the problem. What you described is a dream, but we aren't there yet. We hope that we will be there sometime.
Thank you.
Manon, you can follow with your question.
Yes, thank you for your talk. It's very interesting. I'm not an expert, so I'm going to have a basic question. The data you as input in the model is kind of static in the sense, okay, there's a mutation or there's not a mutation, or there's an interaction and there's no interaction. Is there any way to take into account dynamics or time frame of if it's a network of protein interaction, some of them might be very stable or very transient. Yes. So is this like useful information, and is
I mean, you are totally correct in this. Actually, I mean, there is a lot other than just taking a measurement in a certain point and building your prediction on this point. But taking multiple point measurement actually is something that is very expensive, and usually it doesn't happen in the protocol that we are following. Usually a patient will be sequenced at a certain point of the treatment where we feel that this may be helpful in deciding the trajectory of the patient in the future, like assigning him or her to a clinical trial, or like if the patient is like experiencing like very severe side effects, so we are trying to change the treatment. It happens at certain points, but usually it doesn't happen multiple times in the treatment plan of the patient. And if you'd like to follow up with a certain patient and get his tumor sequence multiple times, this is an expensive process. We have limited information about such patients. For example, I think our group published on a set of patients where we had like timeline measurements for these patients, but the number of patients was like very small, like 15, 20, something like that, right? So you cannot do this on a scale. Usually we do this for like a research project, a small research project that you follow up with these patients over time, and you get multiple samples. And I mean, you think about so many ways actually. It may be multiple samples over time, or it may be multiple samples from different parts of the body, from different lesions in the body, things like that. It is very dynamic, as you said, but also even the measurement that we take. For example, if you are taking gene expression, usually the thing that we do is that we take like bulk RNA-seq, and this is not ideal as well. For some patients, we take single cell RNA-seq, which means that we get the expression of every single cell inside the tumor. This is informative actually to know the differences between normal and tumor cells, immune cells, microenvironment. So it gives you richer information set, but also with a lot of expenses associated with it. So for example, single cell information and for like 10 patients is a lot of information. So you can scale it up as well. As you can see, the more information that you get from the patients, the less number of patients that you will get, and the more expensive it will be. So we are trying to compromise in different directions.
Thank you.
But this is a great question. Thank you.
So I think we've concluded the activity. So I would like to thank everyone who joined and participated in this activity. And most importantly, thank you, Itam, for your time and your presentation. And always, as a quick reminder, if you want to present your work or an article, you can always register as a presenter on our website. Otherwise, you can always register as an attendee. Also, every last Monday of each month, we have a new presentation, and the next one will be on July 25th. So we'll announce the next presentation and presenter the second week of that month on the website. So goodbye, everyone, and have a nice day, and see you next time.
Thank you. Thank you, everyone.
Thank you, Thank you so much.